Welcome, everybody. Good evening. I'm Dr. Michael Stamus, Dean of the School of Medicine here at UC Irvine, and I'm delighted to welcome you all here today for the first of this academic year of our Dean's Distinguished Lecture Series. We started this series in 2019, really with the idea of bringing influential leaders from healthcare and academia to share their insights, their thoughts, their vision, and their anecdotal stories with us in this environment, and really have a little bit of a pause in 2020 and 2021 because of a little pandemic, but we're back in full force now, and we're delighted to have our guest here tonight, which we'll introduce in a moment. But the idea here is really to look at the world of science and healthcare and medicine sort of through three different lenses. First, from the clinical perspective. Second, from the clinical research perspective, that is the translational part, and then, of course, the fundamental part that leads to those great discoveries and advances in healthcare, which is the basic science component. And that's really what tonight's guest and lecturer is all about, really creating, understanding and developing those basic fundamental concepts that will then lead and have led already to great healthcare advances. So, honored to inform you that our speaker tonight is a 2021 recipient of the Nobel Prize and a world-renowned molecular biologist and physiologist. His name is Dr. Ardem Patapoutian, and he will share with us his insight, his experience, and his vision in his presentation entitled, "How Do You Feel? Molecules That Sense Touch." Just a few householding items. We would love to have some questions and Q&A at the end, but please hold them until we get to the end of the talk. We also are recording this because we like to archive these on our website, so be aware that we are recording. So if you don't want to be recorded, probably don't want to ask a question, maybe have somebody else ask it or give it to one of us to ask for you. And then we hope you enjoy the evening. And with no further ado, I'll ask Dr. Jeff Abbott, who is the Vice Dean of Basic Science Research and really the host designee for the Basic Science Lecture Series, and ask Jeff to introduce formally our speaker.
Thank you, Dean Stamos, and welcome everyone to our Dean's Distinguished Lecture Series. I'm so glad to see people here from high school students all the way through to emeritus faculty. It's an incredible turnout, and we're so excited. It's my great pleasure to introduce to you a brilliant scientist and amazing person. Dr. Patapoutian will be sharing with us tonight his presentation titled, "How Do You Feel? Molecules That Sense Touch." It promises to be a truly fascinating talk. Ardem Patapoutian is a molecular biologist and physiologist. His laboratory identified the molecules that sense temperature and pressure involved in touch, pain, and regulating blood pressure. Dr. Patapoutian was born in Lebanon in 1967 and immigrated to the USA in 1986. He graduated from UCLA in 1990 with a Bachelor's of Science and received his PhD from Caltech in 1996. He joined the Faculty of Scripps Research in 2000, where he is currently the Presidential Endowed Chair in Neurobiology and an investigator of the Howard Hughes Medical Institute. He's a member of the National Academy of Sciences, 2017, and a member of American Academy of Arts and Sciences, 2020. He is a co-recipient of the 2020 Kavli Prize in Neuroscience, the 2021 BBVA Foundation Frontiers of Knowledge Award, and the 2021 Nobel Prize in Physiology or Medicine. It is my distinct honor to welcome to the podium Dr. Ardem Patapoutian.
Hello, everybody, and thank you for the invitation and the kind introduction. This room actually has special meaning for me because, 2021 was still in the middle of COVID when I got that phone call from Stockholm, and instead of us going to Stockholm to receive the medal, we actually came here, right here on this podium, where I think the ambassador from Sweden came, and instead of getting it from the king, got it right here from the ambassador. So yeah, I remember practicing getting up and bowing and taking the medal. So very nice memories. So it's good to be back. So yeah, tomorrow I will. There's a great ion channel symposium, and I know a few people are here from there. So today's talk is meant to be a public talk, so hopefully even high school students should understand. And tomorrow I'll give a more technical science talk. I like to start with a picture of the people in the lab, and this is lab reunion that we had just last year. So you can see all these wonderful young scientists who've worked with me over the decades. Some were in the lab right now, some have graduated and become faculty themselves. A few of them even brought their progeny, which we call our scientific grandchildren. So it's just a lovely event to get together with all these people and the work that I'll talk to you about. Another way to show the same thing instead of faces is this map of the world where we've put a pin on the birthplace of every member of my laboratory over the years. So that's me. I'm of Armenian origin, grew up in Lebanon. I came to US when I was 18 years old. And as you can see, we have pretty nice representation from at least the northern hemisphere. We still can work on our diversity for having people from southern hemisphere come and join our lab. And I'm not showing this here, but the male-female ratio from my lab has always been almost exactly 50%, something I'm very proud of. So what is the science that we do in the lab? We are what we call sensory neurobiologists. So neuroscience mainly concerns itself with the brain, where all the information is being gathered and decisions are being made of how to act on them. But the brain can't function without input, and the sensory systems provide that. And we often talk about the 5 general senses of taste, smell, vision, hearing, and touch. And over the last few decades, we've learned a lot about how you see and smell and taste, but this more enigmatic sense of touch that doesn't depend on chemicals like taste and smell do. And we know a lot about how cells communicate via chemicals. This was kind of an uncharted territory because touch, in the bigger sense of it, depends on mainly physical forces. So how do you sense mechanical force or temperature or even chemicals when we talk about itch? And at the molecular level, how we know this was not known. So this is what I'm going to talk to you about today. But to start with just some fun slides to say that we are very, very particular about temperature. So I bet some of you feel like you're a bit cold maybe now. Some might feel like you're hot if you have to run over here. And I like this because this is the ultimate example of how car manufacturers have taken advantage of the fact that we're all very particular about temperature, and they have designed this dual zone temperature control so that the driver and the passenger can enjoy slightly different temperatures. On top of this, if you're like me, love wine, you realize how particular we are about the precise temperature at which we enjoy a red wine versus a white wine, for example. So the other side of touch, I would say the main side of touch, is this idea of mechanosensation, pressure sensing. And most of this has been evolved for our ability to have discriminative touch. And our ability here, we take it for granted, but it's quite remarkable. Our sense of touch extends into the nanoscale, in nanometers. This might not mean much to you if I said it that way, but actually a nanometer is 1/1000th of a diameter of a hair. So that much indentation you can sense if you close your eyes and rub your finger over a surface, for example. But touch is not just about discrimination of indentations, it's also affective touch. This is Coco and Puff, our 2 cats when they were kittens. And this emotional sensation that we get from touch, this affective touch, without it, as we found out during COVID times, something that we rely on for our health. And for the related sense of proprioception, how many people here know what proprioception is? So many of you don't know, and that's very interesting, and no blame being put on you, but it's a sense that we take for granted, but I would argue that it might be the most important sense that you have. So proprioception is sometimes called muscle memory. It's how you know where your limbs are without looking at it. So if I can close my eyes and touch my nose, even after I had one beer, tells you that I have a mechanism to realize exactly where my fingertips are in space. And the way I know this is because I have neurons that sense how much stretching is going on in all my muscles and tendons, and from that I have an exact idea of what space I occupy. So very similar to touch, but it innervates muscle instead of skin. And without it, we wouldn't be able to do things like standing up and walking, for example. And we'll come back to this. But my favorite photograph that illustrates the importance of touch is this one here. So this young man has walked into the Oval Office in the White House and to meet the President Obama, and tells him that his friends tell him that his hair looks like the president's. And Mr. Obama, of course, doesn't skip a beat and bends over and says, "Touch it, dude." So, this is not just about him touching and identifying the texture of the president's hair, but coming back to this effective emotional aspect of touch that's so important for this boy to realize that someone with hair that looks and feels like his is the president of the United States. So, we often say seeing is believing, but here president knows that touching is believing as well. So the flip side of touch is pain, and acute pain we call hammer hitting your finger, or a thorn, in your finger is actually essential for our survival because it tells us of danger. But chronic pain is a major unmet medical need, and chronic pain is defined as pain that has served its purpose but still there. And if you followed the opioid epidemic for the last decade, you realize that we don't really have good medications for this kind of pain, and it's very important for the research to give future, hopefully, answers to this. So I'm going to show you couple of fun anatomy slides, mainly to show you now we're going to go into more the molecular aspect of how we sense touch and pain. And this is a video of the neurons that are responsible for everything that I've talked about. So they reside next to the spinal cord and send processes all over the body, skin, muscle, internal organs, kind of antennas of monitoring what's going on and representing this information to the brain. And this video is going to show the development of a zebrafish, the DRG neurons, during development. And all of these little bulbs that look like little grapes are this group of 10,000 neurons. And these little processes that you're seeing are these DRG neurites that are extending into the periphery to sense the environment. One of the most fascinating things about these neurons is that they're extremely thin, and we'll talk about scale in a couple of slides, but they are actually the longest cells of your body. So most cells are measured in micrometer length, which is much smaller than a millimeter, for example. But these neurons, imagine, I said they're next to the vertebrae. They extend, for example, a process all the way to your big toe, and the other side of it has to go all the way to the brain to innervate and tell it. So this is a basketball player who, if you're over 40, you might remember his name, Yao Ming. And he's, in a bizarre fashion, holding his own DRG neuron. But the point is that this one cell is almost 2 meters long, which is about over 6 feet. So that's quite remarkable. And the job of this particular sensory neurons is simple. In this case, if you're a touch neuron and you are extended and sense things in the skin, you want to sense mechanical force, touch, or temperature changes as represented in the skin. And to do this, you need molecules. which are always proteins that are specialized to sense this information. And this is what was not known. So just in case for those of you who don't even know what a protein is, these are kind of the workhorse in your genetic information is coded in your DNA, and there are about 20,000 genes, and they all make proteins, and they do everything. So if you've heard of insulin, that's a protein that gets secreted and tells the rest of your body about how much sugar you have or consuming. Your skin is made of keratin. You might have heard that. That's a structural protein. And what we're going to talk about today is a sensor, which we call, in this case, an ion channel. So the easiest way to think about ion channels is that they're electrical switches in neurons. And you might have heard that neurons communicate with each other using electrical currents. And so these are the sensors and the propagators of that signal. They're remarkable families. So each cell is surrounded by this lipid layer, fatty layer called the plasma membrane. It's great because it protects the cell, but the problem with it is that if you didn't have anything like these ion channels, then you're not in contact with the rest of the world. You're completely isolated. So there's all these mechanisms of various proteins, in this case ion channels, that sit in this plasma membrane and tell the cell what's going on outside of it, for example. And ion channels at the most basic level are super simple. They do two things. They're either closed or open. So when they're closed, nothing's happening. When they're open, ions flow through them from outside to the inside of the cell or the opposite direction. And they're remarkable biological engines in the sense that they can let about 10 million ions flow through each of these proteins per second. Quite remarkable. And again, a simple way to think about it is that when these ions come in, they change the electrical property of the cell, which then the neuron can amplify and send to the same next neuron and get to the brain. In addition to their very interesting properties for basic biology, many drugs that you might have heard of target ion channels, including those for anxiety, arrhythmia, CFTR, cystic fibrosis, epilepsy, stroke, anesthetics, etc. So many of these ion channels that I described actually respond to chemicals. I mentioned already most cells respond to chemicals, dopamine, or any kind of neurotransmitter receptors. But here we're looking for something very different, and that is an ion channel that actually responds to physical force, either temperature or pressure. In a way, trying to take physics and turning it into chemical that the neuron understands, right? So how large are these ion channels? I just have one schematic to realize how tiny they are. So a grain of rice is about 500 times bigger than a cell. So that dot is about the cell that's amplified here. And then ion channel, I can't even put it on the cell. If it's at the cell membrane, you wouldn't see it with your naked eye because it's 10,000 times smaller than a cell itself. And why is it important to find these tiny little molecules that we call ion channels? We think that just finding how and then mechanistically understanding, you would sense pressure and turn it into a signal that the brain understands. I find it very fascinating. Hopefully you do too. And of course, there is relevance to human health, specifically in pain that I mentioned. But one of the things I want to impress upon you is that in research, one of the coolest things is when you get very unexpected applications to what you're studying. And this is the best argument I've ever heard to support basic science, because when you do research for one purpose, then you realize, wow, this is actually very useful for something else as well. And we'll hear about a little bit of this later on. So I'm going to quickly talk about our early work in identifying temperature sensors, and then I'll focus also on these ion channels we call piezo that are involved for touch, pain, and other forms of mechanosensation, as I like to call it. And then at the end, I think we'll have time to some fun slides that I'll share with you. So all of our work has been centered around the idea of this slide, where we identify these tiny molecules, the ion channels, that are activated by temperature and pressure, and then we do some work in animal models. We use mice, for example, to understand exactly how they work. We also use clinical data from our own species to find out what they are important for basic physiology and disease. So the first ion channel that we identified in the lab is called tryptamate. This was also found by David Julius's lab, who received the Nobel Prize with me. And for the first time, we showed that an ion channel can be activated by simply cooling it. So how do we even know if an ion channel is activated by cold? So what you're looking at here is what we use to electrically record from the activity of a single protein. So the tiny protein that I talked about is in red, and the plasma membrane, the fatty layer, is in blue. And what we can do is a patch come and isolate it and actually record the currents that would go through this single protein when we give various stimuli. So in this case, in this about 1 second that we're recording from, what you can see that we're cooling it from 30 degrees centigrade to 18. This is going from 18, now I'm not going to be able to remember what Fahrenheit is, but this is cold, and this is warm. And so you can see that initially you see single channels opening, so this is closed. You see one channel, one protein open, and then the colder you get, you realize that there were actually multiple channels in this patch because you see 1, 2, 3 steps. Again, just to give you a scale, the y-axis, as we call it, is how many amps, in this case, picoamps, and 15 picoamps, compared to 100-watt light bulb is 1 amp, so this is tiny. Picoamps is 10 to the power minus 12. So you need very sensitive equipment to be able to record these activities. One of the cool things that happened actually is that we found out that tryptamate, this cold activated channel, is also activated by menthol, which is an ingredient in mint leaves. And you might have experienced the cooling effect of these compounds in a variety of over-the-counter products, including toothbrushes, pain rubs, etc. So next time you get that minty, fresh coolness in your mouth, realize that it's a compound that activates the same channel that cold temperatures activate. I'm not going to show you, but we've shown that tryptamate is indeed the sensor that your body uses to sense cold temperatures. Another one of these ion channels that we found is actually interestingly activated by many spicy compounds. So one of them is wintergreen oil that is present in Listerine. So again, next time you use this product and you feel that burning in your mouth and you're convinced that that burning means that you're killing bacteria, you're actually just activating your pain receptors. If you like sushi, wasabi also activates tryptase-1, and raw garlic, another chemical, activates tryptase-1. We've done lots of work on this, and one of the interesting things was that if you're a chemist and look at those compounds on the left, there is no structural similarity. Usually chemicals that look like a certain way bind a pocket in a protein and activate it. In this case, it was a very unusual activation method. The only thing these compounds have in common is that they're reactive. So they're directly binding to this ion channel and activating it. And its general property of it being responsive to reactive chemicals is actually something very interesting, because we think that blocking this might be very useful for blocking certain aspects of pain. And indeed, this is not our work, but pharmaceutical companies have found these blockers, and they're currently successful phase 2 clinical trials showing efficacy of these blockers in various indications of pain, including arthritis. So I'm going to switch now and talk to you about mechanosensation, pressure sensing instead of temperature. And this work started by a postdoctoral fellow at the time in the lab. Now he runs his own lab in Marseille, France, Bertrand Coste. And it's easy to take a cell and cool it down or heat it up. How do you check if a cell responds to mechanical force? So this is a cell, and here's a recording pipette that records electricity. And Bertrand just comes with a glass probe and just touches the cell like this. And if this cell expresses, it means this protein is on, then you would see this mechanically activated currents, what we call it. So by simply pushing on the cell, you would see currents, this electrical activity that occurs in the cell. So in science, there's a concept of being reductionist. So what does that mean? So we were looking for an ion channel responsible for touch and pain. And when we tried to find it from these neurons that I told you about, it was very difficult, and we failed again and again. And then the reductionist idea was that none of these pressure sensing molecules were known. So why don't we go to the most simpler system, which is not an animal, but a cell that we can culture and grow in an incubator. And if it is mechanosensitive, maybe we can find the molecule from there and then go back and check why is it important for physiology and disease. And this is what Bertrand did. Identified the cell that's very easy to grow and that is mechanosensitive. And using a variety of approaches that I don't have time to tell you, he narrowed it down to about 300 genes that he thought could be the mechanosensor. And then he, using at the time a novel technology called RNAi, which simply means you're knocking down the expression of one gene at a time of these 300 candidates, and try to see which one is responsible to see mechanical activation. So I like to show this slide because it's so simple, and yet he worked full-time 1 year on this. So what you're looking at is the current that he measures from this cell, and then he knocks down one candidate at a time, and he measures it again. Each takes about 3 days. And as he's doing this, he's getting what we call negative data, no new information, day after day after day. Finally, when he got to the 72nd candidate, which is 11 months after he started the screen, and he knocked it down, he saw that the current is massively smaller. So this gene was named, when the human genome was sequenced, many of them, we know what they do, their products, so they have names that are more meaningful. Others, they have no idea what they do. And this one was called Unknown Family 38 Member A. And so he renamed it piezo1 from the Greek piezi, meaning pressure. So over the years, we've worked on these molecules more and shown that they form a novel family of ion channels that are mechanosensitive. There are 2 of them in our genome, both in mice and humans. We call them piezo1 and piezo2. But there are homologs, and I'll talk about this at the end, in many different species, animals, plants, even unicellular organisms. And one of the other interesting things about this molecule was that they are massive relative to other proteins. So most ion channels have what we call transmembrane domains. Because it's a lipid, they have to go around the lipid once or twice, sometimes 6 times. So piezo does this 38 times, more than any other protein in our genomes. On top of that, they come together, 3 of them, to form a trimer. So this is, we've solved the structure of piezo1, and this is what the model of the molecule looks like. So each color is one of these isoforms, 3 of which identical units come together to form the protein. So in the middle is where the pore is, where ions would flow through. And all these little helix that you're seeing are transmembrane domains, kind of extended into the lipid plasma membrane, in a way sensing membrane tension. So this is a schematic of what they look like. In blue is this plasma membrane lipid, and these proteins kind of almost dig a hole and form a cup-like shape in the membrane waiting for this cell to be stretched. And when it does, the arms expand and ions can go through. So one of my ex-trainees actually solved the structure of this ion channel with the arms extended, and this is morphing of the two structures. Again, these are nanoscale molecules, but this is what we think is happening in the cell, that when you stretch it, the arms actually come down like this, flattens, and ions can go through. So that's at the nanoscale, molecular side. What we really wanted to know, of course, is what does it do in an animal? And one way we know how to do this is we can specifically delete a single gene in a mouse model system and ask, does this mouse still sense touch, for example? We've done dozens of experiments. I'm just going to show you guys one video of comparing a mouse that doesn't have piezo2 to ones that does. And again, we've done many very quantitative assays, but I like to show this video because it's the first thing we tried, and we didn't know at first how to even ask if a mouse can sense touch. So one of the experiments that my graduate student, Sanjeev Rana, came up with is not a sophisticated one, but he called it tape on the back. So this is a mouse, and there's a white tape on the back, and it doesn't like it, and it tries to take it off. And here's an animal that doesn't have piezo2. It doesn't seem to care at all. And yes, this is what I got Nobel Prize for. So we're coming back to proprioception, this sense of, I told you where your limbs are in space, and if you're really, really talented, you can do something like this thanks to your proprioception. So we had shown that proprioception is also not functional in mice without piezo2. But one of the fascinating turns of events that happened is that Alex Chesler and Karsten Bannemann, investigators at the National Institutes of Health, identified a group of individuals who also do not have piezo2. So there's a very interesting clinical background to this, because these individuals had gone to their doctors, they have trouble walking, they're not coordinated, some need wheelchairs, and most of the clinicians thought there was something wrong with their muscles or their motor neurons. It is only after my collaborators got involved, sequenced their genome, and found out that they have a mutation in a gene that we have been characterizing as a touch sensor, realizing that lack of touch also can give rise to these, in a way, phenotypes. So this is one of these individuals who has what we call a null mutation, which means the protein is completely dysfunctional, and she cannot sense touch, and she doesn't have proprioception. And one of the ways you can test proprioception is she's blindfolded, she's harnessed for her own protection, and asked simply to walk in a straight line. Something that you and I will have a very easy time doing, but as you can see, she totally cannot do this. So how about pain? We know that both in humans and in mice, without piezo2, acute pain, the hammer hitting your finger, is relatively normal. But we were interested in this phenomenon called tactile allodynia. So two complicated words, all it means is when touch becomes painful. So you and I experience this, for example, after sunburn, where just wearing a shirt is painful. But this is also a symptom that people suffering from neuropathic pain experience constantly, and there's, as we discussed, no easy medication for this. So we had showed already that in mice, this tactile allodynia is dependent on piezo2, but the presence of these individuals who don't have piezo2 and they're willing to be tested for this is very important to translate this knowledge. And one of the main reasons pharmaceutical companies have stayed away from finding the future medicines in pain, because they say that what you find in mouse doesn't usually translate to humans, so we're not going to waste our time working on it. And so to show that this same thing that we found in mice is happening in humans is very, very useful. So Alex Chesler's lab designed this very simple experiment where, just like sunburn, if you rub chili pepper on your arm, it inflames and touch starts becoming painful. And so people are, blindfolded, either control or piezo deficient individuals. And after capsaicin, you touch point A and B and say which one is painful. So if you look at the control arm, of course, 100% of people knew that the part of the arm that got capsaicin became painful. When you ask the individuals from piezo to deficient, they said, can't feel anything. And you say, oh, you have to guess A or B, that's the way it works, and they guess, and they're right 50% of the time. So they've completely lost this mechanical allodynia, tactile allodynia, which is pain in response to touch stimulus, which again is very clinically relevant. So I can go on, but we're finding more and more interesting role for the same molecules for things that have to do with pressure sensing. And some of these are very intuitive. Some of these are a little bit of, might be a surprise for you. So I talked about touch sensation, proprioception, and pain. We've also shown that these molecules are responsible for something called mechanical itch. So if you, maybe when you were a kid, just wearing a wool sweater really bothered you, it caused itch, that's mechanical itch, or a bug crawling on your skin, which again could be pathological. This is because of piezo. In addition, we didn't talk about this yet, but many of these neurons that innervate your skin and muscle to give you the sense of touch and proprioception and pain also innervate internal organs. And we're finding very interesting roles of these mechanosensors in, for example, breathing. So you're not aware of this sensation, but with every breath that you take, your lung stretches, piezo channels sense how much they're stretched, and from this they control your breathing rate. And we've shown that without piezo2, this respiration is deficient, and indeed many of the individuals that are deficient in piezo2 have massive respiratory problems during neonatal time, and they have to hospitalize for it. Another sense that you might not think about that you have, what we call is baroreception. So there's many ways in your body to control your blood pressure. One of them is that these sensory neurons actually can detect how much your aorta is stretched, again, another mechanical force, and from this, they control your blood pressure so that if the aorta is too stretched, you reduce your blood pressure. We've shown that piezo 1 and 2 are responsible for this sense. Now, the next one you're all very familiar with because it's very conscious and that you might have not thought about it, but the reason you know you have to go to the restroom is because your bladder is actually stretched and you have a mechanosensor doing that. And again, we've shown that without piezo 2, you don't have this sense, both in mice and in humans. The human subjects that I talked to you about actually have learned to go to the restroom on a schedule 3 times a day to avoid accidents. And there's so many more interesting questions that we're still studying that have not been answered. One of them, I love this cartoon by Christoph Neumann. It represents what you and I experience when we have a very large meal. You feel full. I think every language that talks about this has a very mechanical sense. But if I told you that 99% of research on satiety, which is the sense of fullness, comes from studying nutrient sensing, amino acids, fats, and sugars, we still don't know how much the actual stretching of your stomach causes you to stop eating. So how much you eat, when you eat, diabetes, obesity, how much of this is related to this other mechanosensory role? We don't know, but these molecules give us a way to study this. So how does this research help patients? One of the fascinating things that I mentioned already is that these individuals were misdiagnosed to have motor neuron or muscle deficits, for example. So just the knowledge that they lack the sensory mechanism already has helped some of these individuals, because now we know, because of the lack of sensory, the way to avoid this for them is to use their visual system to compensate for it. So if they actually watch their legs while they're walking, they will be able to do this without using a wheelchair, for example, and some people have already benefited from this. So as you can imagine, most of treatment in health is using drugs that you take as a pill. Given all the important functions that these molecule does, for example, piezo-2 for touch and proprioception, we can never imagine that a small molecule inhibitor of piezo-2 you would take as a pill to block your pain, because it will cause all kinds of what we call on-target side effects. However, many forms of neuropathic pain are very local to your knee, for example, and one can imagine that topical local inhibition of piezo-2 might be useful, and our lab and many others are working towards finding inhibitors of piezo-2. But this is another example that we started looking for touch and pain, but this research took us to blood pressure regulation, breathing, bladder control, and many of these, like hypertension, asthma, and lower urinary tract dysfunction, are major clinical issues that this research could potentially benefit. So I just have a few more science slides. We mainly talked about neuron sensing pressure, but I just wanted to give the idea that many different cell types experience, sense, and respond to mechanical force. Blood vessels experience blood flowing through them, muscles get stretched, red blood cells get squeezed through capillaries, and we're finding out that there is mechanosensation wherever you look. And the role of piezos is growing in different biological processes and diseases that we didn't think about. And this is just a small list of many different processes that depend on mechanosensation, and many collaborators and colleagues and us are finding roles in them. For piezo-1, this includes role in lymphatic vessel development. I don't have time to go through it. For bone, you might have heard that, for example, astronauts who go into outer space come back with reduced bone density. And that's because bones require pressure for them to stay healthy. And it seems like piezo might be one of the ways where pressure sensing is happening in the bone. We've had a collaboration with Patapoutian here at UC Irvine about wound healing and piezo in the skin. And we've done a lot of work on finding interesting roles of piezos in both red blood cells and white blood cells, including a role in malaria that I won't have time to talk about. And as I mentioned early on, this is what we call an homology tree across evolution. So everything I talked to you about is more mammalian and human piezos, but the same molecule in its different form, of course, is present in many other species, including flies, worms, unicellular organisms, including plants. Actually, a couple of years ago, one of my postdocs studied piezo in a plant and found an interesting role. So plants also experience mechanical force. One example is when they send roots into the soil and they experience a harder surface, they have to detect this and decide, is it hard enough to go through? Is it soft enough to go through, or it's too hard and the root has to go around it? So Alireza Mousavi found that piezo version in Arabidopsis, a plant, is actually expressed exactly where mechanosensing should be happening, which is at the root tips. And I won't show you the data, but he showed that without piezo, plants actually cannot penetrate hard surfaces. So I tell this story just because I find it fascinating how evolution, once finding a sensor that can sense mechanical force, use it in us to sense touch, and we'll use it in plants to sense surface hardness. So some take-home messages. We identified sensors of temperature and pressure. These are essential for touch and proprioception, but also have many unexpected roles in various physiology and disease. And also, again, want to emphasize this timeline. You might have even heard this, that, it takes 10 years from basic research to translating something to a drug. It's actually even longer. We identified this TRPA1 in 2003, and there's phase 2 clinical trials started in 2022. And so it's a long process. This is just a slide to thank all the current lab members, alumni, and all the collaborators from all around the world that I've worked with over the years, and funding agencies. I named some of the key players early on, and I promised Michael I'll say Stuart Cahalan, who's a great postdoc in my lab, and mention it. He's one of the alumni here mentioned. Okay, for the last few slides, I want to kind of tell you what it's like to win a Nobel Prize through my own Twitter account. As you can see, I refuse to call it X. So it is indeed true that to announce it, although I received it here, you first hear of it from a phone call from Stockholm about 2 a.m., early day of October. And just like any other normal human being, I had do not disturb on my phone, so I didn't get the phone call. But somehow they quickly Googled and found another Patapoutian in California and called them up. And it was my 94-year-old father in North Hollywood, north of here. For some reason, he was awake at 2:30 in the morning. He first yelled at them for calling so late, but then figured out what was going on and called me, who, your favorites can bother you in the middle of the night, but others can't. So he woke me up, and I literally found out that what was going to happen. I had like 3 missed calls from Stockholm, and a few minutes before the actual announcement, and my wife Nancy got my son, who's 16 or 17 years old at the time, to come up, and together this is the moment where we're watching the announcement. You might notice that he's touching me in a very affectionate, affective touch kind of way, which really, really doesn't happen that often. So this has very, very special meaning for me. Some of the perks for receiving the Nobel Prize is you get a reserved for Nobel laureate parking spot. There's actually interesting story to this, because this is in the basement of our building, so I was happily using this spot when they called me and they said, "We want to give you another spot." I said, "Well, I don't want it. I want this one." They said, No, you don't have to use it. It's for advertising." It's actually going to be on the first floor where people see it, because no one can see it in the basement. So I've actually given it to my lab. Everybody uses it 1 week or a couple of weeks a year, so that kind of works out. Another perk, I got to throw a first pitch at a San Diego Padres game. That was a lot of fun, and then got to celebrate with my lab, as you can see. But probably the highlight was, because I'm of Armenian origin and the first Armenian to win a Nobel Prize, Armenia invited me, went to Yerevan, had a wonderful time, and they even issued a stamp with my face on it, which is quite difficult to thank you. But you get a lot of attention when you receive the Nobel Prize, so I have a committee of people who, headed by my wife, who want to make sure I don't get a big head, and she even tweets things like this, where I'm taking the garbage, and you can see how happy I am to be photographed. Anyways, thank you so much for your attention.
Really impressive and amazing talk. Really, you were able to synthesize all that information, a little self-deprecating humor even, and really just allow us all to understand exactly what you're talking about. So thank you for that. We have time for a few questions from the audience. If we have anybody, we have microphones around, please. Yeah.
Hi, I'm Colleen Cunningham, and I'm a pediatrician. And it's long been known that young infants that aren't touched aren't healthy. Do you think these channels? Do you think the lack of touch, the impact of that is mediated through these mechanisms?
I would say it must be, although, so there's two kinds of touch. I told you about the one that helps you discriminate indentations on your fingers and the affective touch. And so affective touch is a completely different kind of peripheral nervous system, different neurons than the other ones. And so most of what we know from the very few individuals without piezo2 is that they can't discriminate touch. They do seem to have a little bit of affective touch, and we don't know if that's because of another sensor or they've compensated so that they use the visual system to get that benefit. But they say they love hugs. Whether they're feeling it or they're seeing it is difficult to answer. But there's more details too. So touch is almost completely blocked, but strong pressure like a cuff is not. And so there's a little bit of remaining, and that's what makes it difficult to answer your question with very strong conclusion. But I would have to say that, of course, that has to be part of it. I think if I would bet, I would say, of course it is. And that's something that's very ignored. As you know, historically, especially very early infants, now they encourage touching as early as possible if they're premature, and good reason for that. Yeah.
Excellent. Another question up here.
Thank you for a fascinating talk. I'm a system neuroscientist. I wonder what you think about the role of the network, especially the brain, top down, right, in how we feel, especially phantom pain in amputees. So what's your take on the dynamics between your bottom-up approach and perhaps the top-down role?
Absolutely. So I'm sure many of you know phantom pain is this terrible thing where you've lost an arm, but you still feel pain in your fingertips that you don't have. And in the literature, this is mainly thought to be a brain CNS top-down problem because of rewiring or whatever. But there's one interesting tidbit which tells you that both are required. In many of these individuals who have phantom pain, wherever the cut is in the arm, for example, if you use anesthetic at the tip, the pain goes away. So that tells you that, yes, it's a CNS problem, but constant input from the injury site is still important in causing. This becomes more important when you talk about pain relief and pain research, right? Because there's a camp that says its pain is in the brain. It doesn't matter what you think about in the peripheral nervous system. The problem with that, as it is with opioids, is there's going to be side effects because there's not as much specificity there. While in the periphery, you might have the problem of that, it's not going to explain everything, but you have specificity in the sense that, something that senses noxious temperature is not going to have other side effects in the brain. So I think one has to consider both, and there's positive and negatives. And of course, many other questions I get about, if I eat lots of chili peppers and I don't sense anything, is this at the molecule, at the periphery, or in the CNS? I would say most of it probably is in the CNS, but there's also some desensitization at the protein level in the periphery. So I think both would be important.
Another question I see back here.
Thank you for a really lovely talk. That was great. And now I know why I felt so many of those hits in rugby when I was growing up. So discovery by the Charles Cox lab showed that there's auxiliary subunits to these piezo channels. This might be a little bit outside the scope of this talk, but how much do you feel that could change the pharmacology because of piezo? And what kind of implications do you think that discovery has physiologically?
Yeah. Let me first put that question in a context that most people would understand. So I told you that these three arms of piezos function by themselves as mechanosensors, which is true. But there was a recent study showing that there's another molecule that can sit in there and change the properties a little bit. And so that was a very exciting and surprising result. And we don't know the answer to your question because it's just being identified, but clearly it's changing pore properties of the channel, which means that it's probably going to affect pharmacology. And the more interesting question is going to be in what biological process pain, blood pressure sensing, or pathology, it's going to play an important role. And those are all exciting research that's hopefully going to come up in the next few years.
Many of the examples you were talking about in your presentation, excellent presentation, were related to loss of function of piezo. Mm-hmm. And obviously, there could just as well be overactivation of piezo. And I'm wondering if fibromyalgia might be a condition where there might be a case for piezo interaction at some level.
Yes, I didn't have time to talk about, but we have done lots of work on what we call gain-of-function mutations. So again, I mainly talked about what happens if you take this protein away, and there are mutations that can happen in the protein that would make the protein in a way more active. And so there are such mutations in humans for both piezo1 and 2. For piezo2, it causes this developmental disorder called arthrogryposis, which is their limbs don't develop normally during embryogenesis and postnatal development. And in piezo1, which is mainly in important roles in the cardiovascular system, they cause dehydration of red blood cells, which can cause anemia, for example. So fibromyalgia, very interesting, very relevant to pain, obviously. Never been looked at, but it's a very good question. I actually know a collaborator who's very interested in that.
Hi. So you kind of spoke about developmental disorders just now, but I was wondering if because of the mechanotransduction issues, do you see a lot of developmental disorders or like developmental issues in your patients with piezo2 knockdowns and such? I
think the first problem that these individuals have is perinatally with breathing, as I mentioned, and that seems to be mainly because of the neurons that innervate the lung, and again don't sense the stretch. So there's a Herring-Brewer reflex that is if you overinflate the lung, that you have apnea, stop breathing. This reflex is completely gone without piezo2. But that's for piezo2 individuals, that's the only problem. And somehow, once they get through a few years, these lung problems go away, and the main issue left is the lack of functional touch and proprioception and bladder and all those issues.
It's now 7:30, but I think we have time for one last question, and then we will break after this, after a short ceremony, for little refreshments out there, and Dr. Patapoutian will probably stick around and maybe answer other questions for you.
Thank you for the talk. I'm a professor in the humanities, might be a naive question. Is touch bilateral or unilateral? In other words, is there a reciprocity between the toucher and the touchee? When someone is being touched and someone is touching, I mean, are they symmetrical? Are they reciprocals, or can they be felt differentially?
Yeah.
You intended one way, but I get it another way. Is there a way to kind of mediate between multiple wavelengths, or what seems to be a neutral phenomenon?
I mean, this ties it very nicely to the question of what he was talking about, top-down, which is what the brain controls. Obviously, for effective touch, from one individual, it would be very welcome. From another one, it would be very unwelcome. And that is, both of them are going to be activating your same piezo channels. So the rest are all from the brain controlling this. But I would say it's more than the touch and the touchy. It's also very much related to your internal state. And I give you a simple example of this, is that it's very cold outside. You come in, you take a warm shower, it feels great. But if it was hot outside and you were out for a run, you come in and you take the same temperature shower, it's not going to be pleasant at all. It's going to be too hot, right? So that's the same stimulus still, the same channels, but your valence has changed. From pleasant, it became unpleasant. So all of that is on your internal state. So your internal state decides how you react to a stimulus. These are just telling you what's out there. So your internal state could be about your internal temperature. Your internal state could be what you think about the person who's touching you. But there's lots of, brain control over how you do the interpretation of these input messages.
So really, again, an excellent presentation, excellent talk, excellent way of helping us understand this, and I love the way you can even answer what seem like complicated questions and help us understand them. Last time you were on this stage, it was quite a special event. I'm sorry you didn't get to be presented by the king. I'd like to think a dean outranks an ambassador, but I'm not sure. But we do have a small gift for you. I know that our gift and our honorarium will not match the Nobel gift, but nevertheless, we do want to recognize your efforts and thank you. Extremely grateful for your visit to us and presentation. It says here, UCI School of Medicine, Dean's Distinguished Lecture, Ardem Patapoutian, PhD, October 17th and 18th, 2023. This is quite a bit to touch, so be careful.
You just have it.
Take your pictures. I want to also thank a few people who helped make this event possible. First of all, the Beckman Center for this beautiful space, this fantastic venue. UCI Media for recording the video, and it will be available on our website very soon. Thank you. Noah, our photographer for the evening. Thank you. And all the people behind the scene in my office, including Emily Dawidoff, Teresa Waters, Raina Sosha, Claire Bernard Draper, Gina Giuliano, Jim Swenson, Jessica Banfield, Michelle Strombeck, Patrick Rodella, and many others. But also, especially thank you to this amazing crowd this evening who've made this distinguished lecture a real success. So thank you all for being here. Please look to our website for news of our next speakers. We will continue to bring you hopefully similar quality and caliber speakers in the future. That's our goal, of course. And please join us in the atrium outside for refreshments, and thank you for joining us.